All right, let's dive straight into this explainer. Because mastering Cambridge Biology 9700 topic 1.2 is absolutely essential. I mean, centrioles, microtubules, cilia, and microvillia, they show up in almost every single examination series. So today, we're going to walk through exactly what the examiners are looking for, step by step, so you can lock in those top-tier marks.
Now, consider this answer for a second. The centrioles and microtubules moved the cell parts. It seems okay, right? Well, actually, this is a specific painful mistake that costs students those A-star marks every year, usually before they even realize what hit them. You mentioned the organelles, you mentioned movement, but an examiner is immediately going to bump you down to a lower mark band simply because it lacks structural and functional precision.
Here's the thing, reading the textbook 10 times over just won't fix this. Memorizing basic, superficial facts isn't going to save you when the exam strictly prioritizes deep understanding and precise cause and effect. An examiner isn't just checking to see if you know what a microtubule is, they want to know exactly how and why it works inside the cellular machinery. To truly excel here, you've got to understand the three distinct areas you're actually being graded on. Cambridge isn't just testing your memory of microvilli. They're assessing your foundational knowledge, sure, but also your ability to handle complex biological data and your experimental skills. They really want to see if you can apply a core concept to a brand new graph or a totally unfamiliar scenario. This right here is like the ultimate mental map you need. Notice how we aren't just listing the structure. We're linking its core foundational principle directly to a broader cross-topic connection. For example, examiners absolutely love it when you connect the rhythmic motility of cilia directly to respiratory defense mechanisms. That is what shows true connected understanding. So, that brings us to a massive question. What is the hidden examiner clue that flips a low-tier response into a perfect score? It's a tiny detail that changes literally everything. But to spot it, you first need a specific analytical framework. Without it, you'll just be guessing. But with it, the answers practically write themselves. Let's get right into Section 1, the method mark strategy. This is all about showing your systematic process, not just jumping to your final answer. You've got to treat your biological knowledge as ammunition for this simple 4-step framework. First, clearly identify the core concept. Second, integrate any data they give you in the prompt. Third, apply rigorous scientific reasoning. And fourth, wrap the whole thing up using precise examiner wording. This is exactly how you engineer a high mark response. And this builds into a standard sequence you can use for analyzing unfamiliar data or practical work. You identify the key elements, apply those biological principles, execute your analysis with clear working, verify your results, and present it all properly. It's a completely logical sequence that leaves zero room for examiner doubt. Just look at the difference here. It's night and day. Our flawed example is just a vague guess, but the top band response? Centrioles organize the microtubule spindle fibers, which contract to separate chromatids during anaphase, transferring precise genetic data. See what happened there? That forces the examiner to award the mark because you gave the exact mechanism and the biological purpose. Okay, but what happens when the exam shifts from pure theory to data analysis? Your scientific reasoning has to include selecting the correct statistical test. If you're comparing the mean length of microvilli in 2 different samples, you need a t-test. If the data is categorical, go with chi-squared. Paired variables, use correlation tests. Knowing why to use a test is just as important as knowing how to do the math. And similarly, if a question tests your experimental skills regarding cell structures, you must sequentially demonstrate these exact practical competencies to grab those method marks. Explicitly state that you're selecting the appropriate apparatus, recording data in well-labeled tables, identifying anomalies, and drawing evidence-based conclusions. You have to put it all on the paper. This brings us to a real hallmark of a grade A student, evaluative thinking. Top candidates constantly question their own assumptions. If an experiment shows reduced ciliary beating, don't just jump to the first conclusion. Ask yourself, what are the limitations of this data? What are the alternative explanations? Always justify your decisions with evidence. Moving into Section 2, Principal Examiner Warnings. Let's look at the top mistakes that catch students completely off guard every single year. You want to avoid these 3 critical pitfalls at all costs. Number 1, vague answers. Number 2, forgetting to check your units and terminology. And number 3, this is a big one, failing to anchor your biological facts to the actual context of the question. If the prompt is about cilia in the respiratory tract, do not give a generic answer about cilia in a paramecium. Context is everything. Guys, this is a massive alert, straight from the grading room. Candidates consistently confuse key terminology for centrioles and cilia. They mix up the structure, the location, the function, the whole shebang. Your best defense here is to explicitly define your terms before weaving them into your extended analysis.
But here is the terrifying cliffhanger. You could deploy this entire method mark strategy perfectly, know every single structural detail about microtubules, and still score an absolute 0 on a 5-mark question. How? By misunderstanding one single word in the prompt.
Yep, it all comes down to the command words. The hidden cause of sudden grade drops isn't a lack of biology knowledge, it's failing to decode the examiner's instructions. The difference between describe, explain, and evaluate is literally the difference between passing and failing. Decoding those is exactly what we're going to master in our next explainer. So before you jump into your next revision session, ask yourself, are you just memorizing organelle names? Or do you actually understand the cellular machinery driving them? To ensure you don't fall into the command word trap and to keep mastering these frameworks, subscribe to our YouTube channel Ace the Exams at acetheexams_IGCSE. I'll see you in the next explainer to unlock those final mark bands.